Just a splendid talk. Um, my talk is going to be a little, it's going to try to answer two questions. And obviously these questions relate to Charles Darwin, and I will explain exactly how they relate to him in a minute. The, the first of the questions is, the first of the questions is, who are our closest living relatives, and when did we share a recent common ancestor with them? The second question relates to the past and is a slightly longer question. Does our twig of the tree of life consist of just the ancestors of modern humans, or do we also have close fossil relatives that did not make it onto the surface of the tree? In other words, they are extinct. The questions come from the two major ideas in the origin of species. One of the two major ideas in the origin of species. The two major ideas are natural selection, and the other one is the notion of common descent. The tree of life is an image that I'm sure the people attending today are familiar with in ancient literature. But what Darwin was suggesting was the tree of life was no longer just a metaphor, a visual metaphor, it was actual. So the first question is, who are our closest living relatives, and when do we share a recent common ancestor with? And there are various, and so on the left is a modern human. Next to them are the two chimpanzees, the common chimpanzee and the bonobo, and then the mountain gorillas and lowland gorillas. And the two main species of orang, one from Sumatra and one from Borneo. Now, when I was a student, we were told that there was such a major difference between modern humans and all those apes that you put the humans in one family, the Hominidae, and you put all the apes in another family called the Pongidae. But what is the situation as opposed to what was I taught as a student? And there are various lines of evidence that you can use. One of them is what I'm calling traditional morphology. In other words, what do the bones look like? What do the soft tissues look like? And the first person to investigate this was a man called Edward Tyson, who was a doctor. He was medically qualified. He was a member of the Royal College of Physicians. And he was actually the medical superintendent of the Bethlehem Institute for the Insane. And Londoners used to shorten the Bethlehem Institute for the Insane to Bedlam. And that is a word that we use when we think there's much too much noise, and so you accuse your children or the students of creating bedlam. Tyson was a morphologist who was the person in the Royal Society who would be sent organisms that were exotic and should be dissected. And one of the organisms that he was sent in the late 1600s was a creature, and a skeleton of which is on the image there. He didn't know what it was, but it was an infant chimpanzee that had been brought back from Africa by some merchants to Bristol. And he dissected it, and the report of his dissection is a magnificent report. He dissected it along with another anatomist called Cowper. And their dissection was meticulous. And they found that there were very few differences between this chimpanzee and the anatomy of modern humans. The next recorded dissections of a chimpanzee after Tyson were by a man called Trail and by Richard Owen. But it was Thomas Henry Huxley who moved the debate forward when he did some dissections of chimpanzees. And what Huxley did was that he provided additional morphological evidence to that already accumulated by Tyson and Trail and Owen, and in Evidences to Man's Place in Nature in 1863. He argued for a close relationship between modern humans and the apes, especially the African apes. And what he wrote in Evidences to Man's Place in Nature is whatever part of the animal fabric might be selected for comparison. The lower apes, the monkeys, and the gorilla, and there are various reasons why he focused on the gorilla, would differ more than the gorilla and the man. In other words, what he was saying was that the apes and man, human beings, were more similar than the apes and monkeys. So what is the situation today with regards to traditional morphology? Not long ago, I had a graduate student called Rui Diogo, and what he did for his PhD thesis was to look at the morphology of the striated muscles. These are the muscles that move your limbs. These aren't the muscles in your gut or your heart muscle. And when we looked at the striated muscles, and we looked at what they look like in modern humans and in chimpanzees, and latterly in bonobos and in gorillas and orangs, it was obvious that the muscles, the evidence from the muscles, linked the African apes together, gorilla, modern humans, and chimpanzees. So you can see here that the striated muscles in the gibbons, which is the top row, are very distinctive, which is probably not surprising given their very unusual locomotion. And then there are shared features in the common ancestor of orangs and the African apes and modern humans. There are more shared features in the common ancestor of the African apes and modern humans. And there are a few shared features in modern humans and chimpanzees. So striated muscles reinforce the results from Tyson and the people who came after him. So what about the molecular evidence? Well, the molecular evidence was investigated originally by two researchers. One was called Emil Zuckerkandl, and what he did was that he broke a rather complicated protein called hemoglobin down into its component peptides, and then he used a relatively modern method called electrophoresis, or modern at the time, to see if he could separate the peptides. And when you visualize the peptides, when you spray material onto the gel, you can see that the patterns of modern humans and gorillas are almost indistinguishable. But they can be distinguished from the gorilla and the orang, and especially the rhesus monkey. The other line of evidence was pursued by Maurice Goodman, who used what was then the relatively new method of immunology to explore the difference between the albumins of modern humans and the African apes and the orangs and monkeys. And basically, what you have to notice in this, there are wells. There are two wells in the top of each of these, and there is a single well in the bottom. And you put antibodies in the bottom well, and you put the albumins in the top well, and they move through the gel towards each other. And the more of a spur there is in the white line, which is where the interaction occurs, the more of a spur there is, the greater the difference. And you can see on the top right that when you do that between a chimpanzee and a man, and you offer those two, you can see that there is no spur. In other words, this particular assay cannot discriminate between modern humans and chimpanzees. And this was the first, this and Zuckerkandl's evidence was the first molecular evidence that there was a close relationship between modern humans and chimpanzees. However, when you look into the literature, there was another person who investigated this who doesn't really get the credit that he deserves. And this is a guy called Albert Leighton, who used to be known as Albert Grunbaum, but who very sensibly changed his name during the First World War from Grunbaum to Leighton. And that's probably why his work is not as well known as it should be. In 1902, he wrote a letter to the journal called The Lancet, and it's called A Note on the Blood Relationship of Man and the Anthropoid Apes. And what he suggested in the letter, and we now know that he was, and basically what he was doing was a newfangled, what he was doing was using what was then a newfangled technique, which was mixing antiserum with serum, and that was called serology. And he wrote that I find that human antiserum gives a precipitate with all three species of anthropoid apes already named. And to me, this precipitate is practically indistinguishable from that obtained with human blood. In other words, if you mix human blood with human antiserum, or you mix human antiserum with the blood of these apes, and he got the blood from the zoo in Manchester. He was working in Liverpool at the time. And then he goes on to say, occasionally it is seen that the blood of the orang gave a more gelatinous precipitate, in other words, was more different, as compared to the granular precipitate of the other bloods, in other words, the chimpanzee and the gorilla. But this may have been due to accidental circumstances. So he sort of said, it may have been my fault. But basically, this is certainly the first use of serology to compare a modern human with a chimpanzee, a gorilla, and an orang. And it is remarkable that Grunbaum, as he was then known, detected the difference between the reactions of the chimpanzee and the gorilla and the orangutan. He should get more credit than he has been given. And then, of course, Watson and Crick, with the unwitting help of Rosalind Franklin, once DNA was discovered, and you've heard from Mark Stoneking how much it can be used to explore human evolutionary history, more recent human evolutionary history, that confirmed the close relationship between modern humans and chimpanzees. And instead of the diagram I showed you at the beginning, we now know that modern humans and canins, which is the family name, the tribe name for chimpanzees, likely share the common ancestor between 7 and 6 million years ago. And then a little more distantly come the gorillas, and then even more distantly come the orangs. So if we go back to our question, who are our closest living relatives? Um, the answer is chimpanzees and bonobos. And then the other question, when do we share a recent common ancestor with them? Um, the answer to that question is a little um, less precise. And some people argue that the common ancestor was older than 7 or 6 million years, and some people argue that it might have been a little younger. But I'm just going to use 7 to 6 million years as a reasonable interpretation of the evidence. So that's the first question. Second question, does our twig of the tree of life consist of just ancestors of modern humans, or do we also have close fossil relatives that did not make it onto the surface of the tree of life? Now, we have to have ancestors, but we don't have to have close relatives. I'm an only child. My father was an only child. So you don't have to have close relatives. This is a diagram of what is currently understood of the hominin fossil record. This is a pretty speciose interpretation, and the vertical axis is time. And each of these columns is a hypothesis about the existence of an extinct, or in the case of modern humans, we are still narrowly around, but the rest are extinct. The heights of the columns reflect the difference between the first appearance date at the bottom and the last appearance date at the top. As I said, this is a rather a speciose interpretation of human evolutionary history, but I think it's worth our consideration. The colors are an attempt to try and divide all the extinct ancestors or close relatives into grades. And the ones that are red are modern humans. The ones that are blue have a sort of a body plan, which is more like a modern human than like an ape. The ones that are the orange color and the brown color, these are extinct hominins that have very large postcanine or chewing teeth. And the green ones at the bottom are taxa that have been suggested that might be the first hominin, the earliest hominin. And then the light blue ones are hominins that are neither like the blue ones, in other words, they don't have a body plan, which is more like a modern human than an ape, but nor do they have the large teeth that are characteristic of the orange and the brown ones. Now this, in order to talk about the second question, I'm going to talk about not my evolutionary history as a member of Homo sapiens, but my evolutionary history as Bernard Wood. And so here is a picture of my great-grandparents' golden wedding. And this was a picture which was taken at my father's parents' home in Stoke Newington in North London in 1933. So this is my evolutionary history, not our evolutionary history. Now, when you look at that picture, on the left is my father's father. To his left, the one which is the guy with the spectacles, that's my father. To his left, to your right, as you're looking at it, is my father's mother. And then the two venerable people either side of the very exotic decoration are my grandmother's parents. So they are all my ancestors. They were necessary for my existence. The lady who is labeled with an R is a close relative. That's my Aunt Kitty. She was a delightful person, and I'm very pleased that I was related to her, but she wasn't necessary for my existence. And so in this picture, the ancestor to non-ancestor ratio is 5-1 in favor of ancestors. This is a picture of my christening party, which occurred at a date that I'm not going to share with you. And this was at my father's parents' house in North London. Now, when you look at here, there are some ancestors. There's my father's father on your left, and there's my mother, who is the woman with the A and the glasses. And then to the right, you will perhaps recognize my father's mother, and then my father's mother's mother from the golden wedding picture. So there are ancestors in that picture, but the bulk of people are either close relatives or my godparents, who are not related to me at all. Um, so, um, so in that picture, the ancestor to non-ancestor ratio is much more equal. So let's go back to the diagram of the fossil hominins and inquire whether that is more like my great-grandparents' golden wedding or more like my christening. In other words, are all those columns, all those hypotheses about the existence of an extinct hominin, are they all our ancestors or nearly all our ancestors? Or are most of them close relatives and just a few of them our ancestors? And just to remind you, you must have ancestors, but you don't necessarily have close relatives. And when we look at those columns, you have to interpret the columns on the understanding that the columns are our present understanding of the earliest evidence for a fossil at the bottom and the most recent evidence at the top. It's racingly certain that we will probably find more ancient examples of each of those columns and more recent examples of each of those columns. And so those columns are going to expand in height. The more work people do to explore and look for new fossils. So basically, from just over 4.5 million years ago to very recently, there has always been more than one hominin somewhere in the world. And so if there's more than one hominin somewhere in the world, only one of those can be our ancestor. I mean, that's just the way it works. So, does our twig of the tree of life consist of just of the ancestors of modern humans? And the answer to that question is no. I'm sorry. Do we also have close fossil relatives that did not make it onto the surface of the tree? And the answer to that is very much yes. And it's more like an extreme version of the ratio at my christening than at the golden wedding celebration. In other words, most of the fossils that we recognize in the hominin fossil record are likely not to be our ancestors. So is it easy to tell close relatives from ancestors? Now, I've been very definitive about telling you which of the people in those images are my ancestors and my relatives, because I have evidence, which is, there is written evidence. But in the absence of that historical evidence, how do you tell close relatives from ancestors? And the answer is it's rather difficult if you're trying to use the skeleton to answer that question. So so in theory, reconstructing phylogeny should be pretty simple. The more morphology you share, the more evolutionary history you share. But there is a problem. And the problem was identified by this guy called Eric Lancaster in 1870, who was the director of the Natural History Museum. And he wrote, when identical or nearly similar forces or environments act on two or more parts of an organism, the resulting correspondence is called forth. And don't forget, he was a Victorian, so this is very flowery language. The resulting correspondence is called forth in the several parts of the two organisms will be nearly or exactly alike. In other words, two closely related organisms exposed to the same environmental challenges are likely to respond in similar ways. And he says, I propose to call this kind of agreement homoplasis or homoplasy. Homoplasis never caught on, and in fact, homoplasy didn't catch on for a long, long time. This was 1870, and basically his observation was buried, and nobody really took much notice of it until molecular evidence came along, which was able to give us much more secure information about relationships. And it was only when people realized that morphology sometimes clashed with the molecular evidence that people rediscovered homoplasy. So think of homoplasy as noise that obscures the phylogenetic signal. How do you identify homoplasy? That's another talk. So I would like to thank all my students and postdocs and my colleagues who've contributed to making me seem a much smarter scientist than I really am. And I want to reiterate my thanks to the organizers. And if there's time, I'm happy to answer questions.